Hello and welcome to our webinar on tips for teaching biology for the IB Diploma. I'm Tamsin Hart from Cambridge University Press, and I'll be hosting this webinar for our speaker, Brenda Walpole. Thank you for joining us today. I'm just going to give attendees a little time to join the webinar before we start. If you'd like to chat to one another and let us know which country you're joining us from, please feel free to use the chat box. It would be great to hear from you. While you're waiting, I'm going to launch a poll to find out about your main challenges in the classroom. It would be very interesting to see what answers we get. If I haven't included your main challenge, you can add this to the chat and see if your fellow attendees have similar challenges. So let me pull that up for you now. So I'm just going to end the poll, give you 1 second to get in your answers if you'd really like to, because I can see that surging up to 76%. Okay, I'm going to end the poll now. I'm going to share the results. So what are your main challenges in the classroom? I can see that coming out on top there, we've got preparing my students for assessment, 63% of your choices, 50% applying effective teaching approaches. And then we've got a few, one more after that, 38% planning my lessons. Then we've got some tied answers, following IB diploma approaches, handling syllabus changes, and lack of time management. Those seem to be the main issues. There are others coming in after that. Those are really interesting results. Thank you very much for participating. I'm going to stop sharing those now. Okay. So that is the end of our introduction. Thank you very much for joining in the webinar today. Let me now hand over to Brenda.
Hello everyone, thank you so much for joining us this afternoon. I hope that I'm going to be able to answer some of those questions and challenges that you have indicated in the poll that Tamsin has just done. The webinar is about the new IB specification, the changes in it and the challenges it presents to us as teachers and educators. Before we begin, I'll just say a couple of words about myself and the team who've prepared the resources for Cambridge. I'm the coursebook author. My career has been spent in science communication in education at the Natural History Museum in London, the University of Hong Kong, in the Middle East, and also here in the UK. I'm a former lead teacher at a top IB school here. The workbook has been written by another IB teacher who has 15 years experience and who's worked in international schools in both the UK and the Far East in Singapore. And the teacher's resource, which I think is going to help quite a number of you, is compiled by an IB teacher who's also very experienced and is currently working in the UK. So what are the aims of the webinar? Well, first of all, to summarise the changes to the new IB syllabus for the examinations that will take place in 2 years' time. And more importantly, to outline how Cambridge University Press resources will support you, and to discuss how using the resources can enhance learning for students as they study the course. As Tamsin has already mentioned, there'll be an opportunity at the end for you to ask questions, which I'll do my very best to answer for you. So let's start with the key changes to the syllabus. I'm sure that many of you will already have seen the new specification, but I thought it would be useful to highlight a few of the main changes. Perhaps the most important change is that there are now no options in the new syllabus. Everything is covered in the core and everything should be covered by every student. The approach has changed somewhat. We are asked to be more conceptual. There should be less content to cover. Well, that remains to be seen. The teaching hours are 150 hours for standard level and 240 at higher level. The content, known as the roadmap, is arranged in 40 topics in 16 nodes. But the teaching of these topics is fluid and we're encouraged to select a route that suits our students and suits us best. This, of course, has its own problems and complications. Let's have a look first at what's new or more important. In my view, More important and more important in the syllabus is membranes, membrane transport, communication, both chemical and nervous, between cells. Water potential, certainly at higher level, is now more complex. As one might expect, there's a lot more on defense against disease, particularly in the light of the recent pandemic. And there's considerable emphasis now on biodiversity, conservation and protection of living things, stability and change in ecosystems, and the consequences of human activity. Climate change also has its own topic. As far as assessment goes, the internal assessment can now be a collaborative project. The idea here is that some schools don't have sufficient equipment or resources for every student to do an individual investigation. I would add a rider here that if data is collected collaboratively, it's important that students analyse and produce their own analysis and results from the data they may have collected in a group. The personal engagement mark has now been removed from the project, and as have the specific ICT requirements. We are awaiting the IB's ruling on what statistics they require students to use as they work through the internal assessment. So what have we lost from the syllabus? Well, you may have noticed that obviously the options are gone, but certain aspects of those options are now included in the core. Some that spring to mind are muscle structure and function, previously in the option and now in the main part of the syllabus. The digestive system has completely gone. The raison d'etre for this is that most students will have studied it at an earlier stage in their scientific careers. However, we are still required to use ingestion and egestion as examples, and also to describe peristalsis. So you'll be pleased to hear that digestion hasn't disappeared completely. The detailed structure of the kidney is now removed from the main part of the course, but there is quite a bit to cover on osmoregulation. Also gone are monoclonal antibodies, cloning and gene transfer, the Human Genome Project, and applications. Many of the applications in the old course are gone. So stem cell use in medicine and the use of microorganisms for human insulin production are no longer included. Quite a bit of the historical material has gone. Harvey's work on blood and circulation, Florian Chain's work, the eradication of smallpox are among some of the historical aspects that are now gone. So let's have a look at the new syllabus roadmap. It aims to integrate concepts, content, and nature of science as we proceed through. The first column shows you the themes: unity and diversity, form and function, interaction and interdependence, and continuity and change. Along the top rows, you see the levels of organization. So we can consider each theme either at the molecular level, the cellular level, the level of the whole organism, or at the ecosystem level. If you're able to see, the topics which are in purple are higher level only. So there are certain aspects which are not for standard level students. So we're asked to select our own routes through the content to suit our students and our own interests and instincts. Let's take a look at some options here. You might start at levels of molecular organization with water. Describe the structure of water, nucleic acids, proteins, carbohydrates, and so on, enzymes and metabolism, and proceed to cell respiration and photosynthesis. Or you might choose to deviate once you've covered the basics of molecular biology and talk about cell structure, membranes, and so on. That's the red arrow on this diagram. Or, depending on your students, you may prefer to start at the whole organism level with gas exchange, transport, defense against disease and reproduction. Depending on the seasons in the countries where you're teaching, it may be more convenient to start with ecosystems if the weather permits outdoor field work, in which case you might like to choose the fourth column, ecosystems, and proceed down that route. In writing the course book, this has presented me with a few problems. Obviously, a course book is linear, whereas your teaching may well be quite different. We've divided the course book into 4 units, which can correspond to the 4 levels of organisation, so cellular, molecular, and so on. And in general, the chapters follow the themes of the course in most cases, although not always. So chapter 1 covers molecules and water, chapter 2, metabolism, respiration, and photosynthesis, and so on. Here is a vague outline of how the chapters flow. So the blue chapters are chapter 1, the green chapters chapter 2, and orange chapters 3 and 4. As you can see in writing the course book, I've gone from DNA to gene expression and inheritance. As a teacher and educator, this just seems more logical to me than returning to it at a much later stage. But as I've said, it's entirely up to you which route you choose to proceed through the material. In the course book, you'll find lots and lots of cross-referencing, so it should be easy to find the topics that you want to teach. So what else is new? Well, I would say one thing that is very different is the fact that certain species are used as examples in the syllabus. These two, the black-throated loon and the ringed seal, are 2 examples which the syllabus suggests we use to talk about how organisms survive in and on water and the adaptations that they have to do so. Other new examples include spruce bark beetles, which are affected by climate change, Indonesian rainforests, which have been severely affected by global warming, cyclical succession, and the fragile ecosystem of the coral reef, which again is affected by raising carbon dioxide levels in the atmosphere. A couple of other new examples, which are rather nice, I think, are speciation in bonobos and John Endler's work on natural selection in guppies. When you teach these topics, do check whether a species is actually specified as the example or whether it's suggested. Clearly, when examination questions are set, if John Endler's work is required, which it is, you have to be sure that you use him as an example for your students. So let's look at some more of the features in the course book which should help you. Obviously, there's plenty of content, all cross-reference with every topic. At the start of chapters, we have objectives. Nature of science, which is very important, I think, in the new course, is included. How we understand all the things we do about science. And science in context. Now, this is a nice one for possibly for your less reluctant students. The how we use what we know in biology in the real world. The second row here are really features which are for the students. Test your understanding questions, contents checklists, exam style questions, and linking questions. And I'll show you a little more about those in just a moment. So at the start of every chapter, you'll find learning objectives. These are summaries of what is included. In addition, there are a couple of guiding questions which pull out the most important points of the chapter. Vocabulary points, key points, are terms and terminology which is important that students understand and remember, and they can glance at these if they're not quite sure. Stem cells, which have far more coverage in the new course. Students are required to know the differences between totipotent, pluripotent, and multipotent. So the key points will help them pick out those terms when they need to. Nature of science, again, very important aspect of the IB philosophy. And you'll find throughout the book there are these purple boxes which discuss a nature of science feature. This one's all about margin of error. And students can read about the difference between systematic errors and random errors, and a little bit about how they can minimize both types of error. Very important when they're carrying out their own investigations and experiments. Test your understanding questions. These are threaded throughout every chapter. They're short one answer or short answer questions, just so that students can check as they're reading that they've picked up the main points that are covered in the chapter. Here are just a few from a couple of different chapters. And you get the idea of the kind of thing. Outline special properties of stem cells, name 4 hormones that you would find in the menstrual cycle, for example. Also featured are exam tips, just useful little hints for students. The number of students who, when asked to make a comparison, fail to do so. So remember to use terms such as whereas or on the other hand or but when you write written answers. And picking out some key drawings that might be required. Draw a molecular diagram to show peptide bonds, and remember it's condensation, so water is produced. Science in context. This is the feature I mentioned earlier about how science is applied in the real world. Botox. Most people have heard of it, but do they know what it is? As biologists, we can confirm it's produced by a bacterium and is one of the most toxic substances known, but used in the cosmetics industry, first of all as a medical treatment to treat squint. It has been used to reduce the appearance of wrinkles, and that's very nice, isn't it? Another one here, lymph nodes or glands. Swollen glands are what everybody might say, but as biology students, students should understand that lymph nodes are what glands actually are in this case. And there are many other science in context little features throughout the book like this. Here's the summary checklists that I mentioned. There are objectives or I can statements, a place that will locate each statement in the textbook, and needs more work, nearly there, or I'm fine. By self-assessment is a very important part of the IB philosophy, and this is one way that students can begin to take responsibility for their own learning and assess just how much they've understood and how much more work they have to do. At the end of each section, there are linking statements. Now, again, as you proceed through the roadmap, there are many different turns and connections you can make. In the specification, you'll see that the IB has actually made some links for you. Here are some different ones. Some are from the syllabus and some are those which I have made. But there are many other possibilities, and we're encouraged as teachers to make our own links as we wish. Again, another aspect of the IB philosophy, reflection and evaluation of learning. At the end of every chapter, you'll find a little reflection box. Reflect on how our understanding of genetics has changed. Oh, and could you explain genes and evolution to a scientist of 150 years ago? And again, can I explain the importance of categorizing organisms in ecosystems even though the categories are only rough and ready. So reflection and evaluation, a nice IB feature there that we have included so that it gives you a start to encourage your students. As well as the course book, there's also a digital edition that will come with the course book. This contains mock specimen papers with mark schemes, lots of exam style questions and answers, data-based questions, and answers to all the questions that are in the course book. In addition, there's a detailed guide to addressing the internal assessment, the student's personal investigation, and some exam guidance too. So I thought it would be useful just to briefly look at the assessment, what the main changes here are going to be. Obviously, because there are no options, there are now only 2 examination papers at the end of the course. At standard level, these account for 80% of the students' marks, with 20% being covered by the internally assessed investigation. This is the component that you as a teacher will mark and which will be externally moderated. The format of the papers isn't terribly different from what we're used to. Paper 1 is multiple choice questions, and paper 1B, data-based questions that are syllabus related and address all the themes. That's the 4 topics in the left-hand side of the roadmap. Paper 2, similarly, data-based question, a much longer question based on information that the students are given, as well as short answer questions. And then for standard level students, 1 extended response question. Higher level looks pretty similar, longer examination papers, more marks, 4 database questions in the first paper, addressing all themes, and again, data-based and short answer questions on paper 2. As you can see, the division of marks is pretty similar to what we have for standard level and not significantly different. The IB have given some responses to some of the questions that have been asked about the new assessment. We're assured that the number of hours in the syllabus guide is not an indication of the level of importance a topic will have in exams. So, for example, if you spent 4 hours teaching the details of photosynthesis and 1 hour teaching food chains, it doesn't mean there will be 4 times more questions on photosynthesis. The approximate number of multiple choice questions is 7 per assessment objective for standard level and 10 for higher level. Just to remind you, the assessment objectives that I'm referring to are demonstrate knowledge of, understand and apply knowledge, and analyze, evaluate and synthesize. Now, I've talked quite a little bit about linking questions. And as I've said, linking is very important, but there will only occasionally be linking across themes or concepts in the exam papers. If you look at the specimen papers that are already published on the IB website, I believe, you'll see that linking is not required to any of those questions. It's more a teaching tool that they want us to use to ensure that students realise the importance of looking at living things in their entirety, not just as topics. Another question which has been asked, and this is the response, in the previous syllabus we were asked to carry out certain practicals as compulsory practicals. This requirement has been removed. So, for example, if there are questions in the examinations about potometers, students will not necessarily be required to know what a potometer is, but they will be expected to analyse data from the experiments, given sufficient information to do so. Right, I think that's all I want to say about the content of the new syllabus. I thought now it'd be quite useful to cover a few tips for teaching it. I was going to start with nature of science, just to give you a few pointers on aspects that I think are important and that you might like to cover. Here are 9 of the aspects that the IB list: evidence, observations, experiments, measurements, global impact of science. To teach this as a separate entity would be impossible. So it's very important that you teach nature of science as you proceed through teaching your normal course. Here's one example from the book. New techniques enabled new discoveries to be made. Cytoskeleton features in the new course, but nobody knew that prokaryotic cells had a cytoskeleton. It wasn't until different methods of cell imaging came along. So that's one place where you might like to talk about new techniques. Microscopy is another, obviously. Here I've included a reference to further reading if this particular topic happens to interest any student or teacher, come to that. Gathering evidence. This is a nice one, which is fairly easy to cover in your teaching. If you're going to measure something, it's very important to use the correct equipment and technique. So you can use your eye for estimates, but if you want to measure something more accurately, you might need a hand lens. Something smaller, you might need a microscope or even an electron microscope. Using the correct equipment is very important, and as students carry out their own experiments, they will need to be aware of this. Hypotheses. Again, as they prepare for their internally assessed experiments, students will have to ask a question, find out about whatever it is they're going to study, try and answer the question, draft a hypothesis, and then experiment, and if necessary, revise the hypothesis in the light of their results. You'll find examples of these things in the teacher's resource and the digital content, guidance for the students how to proceed when they've had an idea and where to go next. What are their observations, their literature search, what's the problem, and how can they develop their hypothesis before going ahead to collect data. Science is a shared endeavour. This is another nature of science topic, as is fighting a new virus. COVID-19 features in the syllabus quite extensively, as you'd expect, in discussing viruses and so on. Here, I've included it as a nature of science topic, international mindedness. This is discusses how scientists came together in 2019 and 2020, and working together they managed to mobilize all their skills and develop a vaccine for COVID-19. Many other projects were put on hold, and all sorts of things such as academic credit was set aside. And obviously communication was so important here, and it's worth emphasizing how this sort of thing could not have happened in previous generations. But maybe COVID-19 wouldn't have happened either. Right, so a little more about the Cambridge University Press resources. I'm going to say a little bit now about the teacher's resource. I note from the survey that we did at the beginning that a lot of you mentioned planning new lessons. The teacher's resource has done quite a bit of this for you. Let me show you some examples. Planning. Each topic has been addressed by Helen, who wrote the teacher's resources. It gives you the content, the timing, and some of the resources which we can provide. So, for example, here's DNA replication. It's in subchapter 3.1. It takes about 2 to 4 hours to teach. And you will find resources in the course book where there are a test your understanding questions. You'll find more help in the workbook, which is I'll talk more about in a moment. and also in the teacher's resource as well. The teacher's resource includes PowerPoint slides, tests, and worksheets. So how does the teacher's resource support students? Well, another feature of the teacher's resource is a little table which highlights misconceptions students might have, how you can identify their misconceptions, and suggestions about things you can do to help students overcome their difficulties. As well as those features, we also have ideas and activities. To start lessons, there are quick starter ideas. This one, memory aids, a 10-minute activity supported by PowerPoint slides, which helps students remember complementary based pairing. So, for example, apple tart and chocolate gâteau, or students could share their favorite ideas, popular ones displayed. Then main teaching ideas and activities. This one describes gel electrophoresis, which we estimate would take about an hour to teach. You'll get a list of resources and where you can get them, a description of the activity itself, and also, while students are waiting, suggestions of questions they could complete from the workbook or from the course book. Vocabulary support. Many of us teach students who are not working in their first language, and most students struggle with technical vocabulary, subtle differences like the difference between intra and interspecific competition. The teacher's resource contains activities such as word games, crosswords, and other activities that you can use just to help support your students with their vocabulary. At the end of the lesson or the end of the topic, there are plenary ideas, little role plays or mind maps, short 10-15-minute activities that help students reinforce what they've learnt and help you get an idea of how much they've understood. For assessment, as I've mentioned, there are end of topic texts for each chapter, exam style questions in the course book, workbook exercises with questions in all the different exam styles, which students can either use for self-assessment or you can use as teachers to assess their work. Digital material, as I've said, has more exam style questions and all the answers. So let's have a look at the workbook. This workbook, which accompanies the other two features, is intended for student use, but teachers may also like to use it to refer students to certain activities or questions that they'd like them to cover. Here are a few examples. This is the style of question that you might find, tables to fill in, identifying terms, little hints and tips as well to help students as they learn. Some data analysis questions, this one on action potential, gap fills, fill in the sentences to help students reinforce their learning. And another one here on food chains and food webs. And here, some tips. When addressing command terms, evaluate, include supporting evidence. Another tip, for complex drawings, draw in pencil first. And food web diagrams only require the names of the organisms. You don't have to draw each organism. While we're talking about names of organisms, I don't know whether you've noticed, but the IB does not have any requirement for students to remember the Latin specific names of organisms. They're very happy for the common name to be used in exams and so on. Right, last but not least, implementing inquiry-based learning. So the IB wants students to have a central role in their learning process, not just learn facts. Our job is to support students and try and engage them by triggering their curiosity. I've just got a few little things here which I hope you might find interesting. Develop a learning activity that promotes conversation and allows them to explore aspects of the curriculum. A graph. This is a graph which shows world population with and without synthetic fertilizers. What will happen to world population? All kinds of topics can be discussed from this. Well, human population, for one thing, the effect of fertilizers on the soil, what would happen if we stopped using synthetic fertilizers, and so on. While we're looking at a graph, can I also highlight to you a very useful website? Some of you may know it already. It's called Turner's Graph of the Week. It's a wonderful resource which each week produces graphs for analysis for students. Enormously valuable practice for them for the IB. Recent ones have included COVID graphs, pollution graphs, graphs on the amount of air travel people undertake, and graphs about the extinction of different species. Not just line graphs, but bar graphs and all kinds of other presentations of data in different forms. I think you will find this a useful resource, and they have a huge back catalogue you can access. Other ways of getting students to talk and think. Newspaper articles. Certainly issues about the ecosystem and what humans are doing to it feature large in many newspapers, journals, and websites. These are great starting points for conversations and discussions. And after a class discussion, you can easily send students off to do their own investigation, their own project work. Other alternatives include photographs. Here I've suggested diatoms. What are they? What are they doing? How important are they? What are they for? Or you could hand an object round. A skull, this sheep skull. Ask students, what is it? What's it doing? What are those teeth? Why are they like that? What's the effect of having the eyes at the side of the head? So many questions you can ask about the structure and functions that you can observe in a biological specimen. What other tools have we got? Well, practical tools. The IB proposes three main things that they call tools to assist us with practical teaching and to prepare for the scientific investigation. They suggest experimental techniques, which you'll find in the coursebook and teacher's resource, applying technology, and mathematics. I think we're all very familiar with carrying out practical work, and technology, generating and presenting data. Well, start with Turner's Graph of the Week or generate some data from the internet. And the third one, which is often a problem for biologists, is mathematics. Here are just a couple of examples of using those tools in the application of skills. So students should make measurements for determination of rate of cell respiration. So which of the three tools could you use here? Well, you could get them to measure mass and time with accuracy, remembering to take several readings and take an average. They could show graphs of the data, which is applying technology. And the maths tool could be calculating a rate from their graph. The second example I've chosen is students should use transect data to correlate the distribution of plants or animals with abiotic data. This one would be the maths tool, understanding direct and inverse proportionality, using random or systematic sampling. And also you could include tool 1, which would be making careful observations and counts. So I think I've probably come to the end of all I want to say. I'll just summarize by talking about how the Cambridge resources and the team that have produced them aim to help and support you in your teaching. As I've said, the course book covers everything that you need to know. It has activities and support materials for students and teachers. It has content objectives, which are linked directly to the teacher's resource to help you prepare new lessons and understand all the changes. And the workbook provides activities and lots of questions for students to use or for you to refer them to for support or exam preparation. And the online digital material gives you all the answers to all the questions in the course book, as well as detailed information on how to prepare your students for the new internal assessment. So here we have the workbook written by Alice Tully, the teacher's resource written by Hélène Bonsall, and the course book, which I wrote, and the digital course book, which is available, and the e-book. So I think that's the end of my presentation for now. So I'm going to hand you back to Tamsin, who is going to address some of the questions which I'm sure you've presented.
Thank you so much, Brenda, for that fantastic presentation. It's very much appreciated. So on to our question and answer session. So if you have any further questions for Brenda, please can you put those in the Q&A box, which you can find by hovering your arrow either at the bottom or the top of your screen. And if you've got a smaller screen so it's not showing, you'll find the three little dots that say more. And if you click into that, you'll find the Q&A. Right, let me pull this up for you. Okay, so we have a couple of questions here. So let's start with this one. Please give us more clarification on science in context. So if you could talk about that, please, Brenda.
Yes, science in context is not a requirement of the IB. It's just something which we at Cambridge University Press thought would be relevant and interesting for you and your students. It's applying biological principles to the real world. So as I said, Botox is one aspect, and there are many others throughout the book which just use science in the real world to make students realize it really is important to know and understand what they're learning.
Brilliant. Thank you, Brenda. Now, our next question. Were there any amendments made to the IA specifications in the syllabus?
They're pretty similar. I mean, you can obtain the IA specification now on the IB website. But as I said, the main ones were the exclusion of the personal engagement mark, which was a rather tricky one, I think. You know, a student had to try and convince the examiners why they wanted to study whatever it was they were investigating. So that has now gone. And as I said, the other significant change is the ability of students to work in small groups to collect data. This is mainly for schools which don't have a lot of equipment or aren't able to give students a lot of lab space. Students can work together and collect their data, but they must analyze it as before. I would say that the changes to the IA, apart from the change of name, which it's now called an experiment, are not significant apart from those two things.
Brilliant, thank you. Now, my next question, this is about how do you feel about the amount of content? Does it really seem less than last syllabus?
That's a very tricky one. And the IB have been challenged on this. It looks less, but I think that it's unlikely to be so. The total teaching hours for standard level are supposed to be 150, and for higher level 240. I think it will still be quite a squeeze to teach the whole course in the time allocated. But some of the more historical stuff has gone, and some of the more obscure things from the options have gone. So maybe it will be easier, but there's still a lot of content to cover in the time, is my opinion.
Brilliant, thank you. And here's another kind of exam-based question. So are there any tips for students to attempt the exams?
Well, I think as always, as we always say, look for the command terms. In the digital material, you'll find quite a lot of information on the command terms and how much you're supposed to write for each one. So state, for example, is very different from discuss or evaluate. And if students are schooled in how to recognize those command terms and write the correct amount for their answers, that's one thing. And as far as multiple choice questions go, practice, practice, practice. Look out for the double negative questions. Be prepared to realize that 2 of the answers in any multiple choice question you can remove immediately. And if you don't know, 50-50, it's worth a guess. You have a 50% chance of being right. As far as the data analysis questions go, I think all you can do is give your students plenty of opportunities to look at graphs, analyze data. As I mentioned again, the Turner's graph of the week is a good place to start. They have to evaluate there. Good luck.
Brilliant. Thank you, Brenda. I think we've got a few questions that come through in the chat as well. Do we have any tips on how to effectively apply IB approach in science to students who are not yet proficient in speaking English?
That's always a difficult one. English as an additional language is hard, but no matter what language they speak, they can share and assess how they're doing and evaluate their own progress. All I would say is to get them involved, encourage them to have a go. It is a difficult one. I've taught many students who have English as an additional language, and I think the technical language, give them vocabulary sheets. As I've said, in the teacher's resource, you'll find lots of word games and things like that, which you can give them to help support their language development. And it is amazing how quickly it progresses once they get underway with the course.
Thank you, Brenda. Now, let me see, what have I got? I've got some more in the chat. Any suggestions about the prior knowledge and skills that students are expected to master? For example, is Cambridge IGCSE sufficient as prep?
Yes, actually, Cambridge IGCSE is excellent preparation, as is the middle years programme, which many schools also do. I think it's a question of being prepared to take risks, have a go, speak up, ask questions, and think about how and what they're learning as much as the facts themselves. There are comments about topic-based learning, which is fine, but I think we have to remember in this context that there's one examination at the end, so we mustn't let topic-based learning take over from the fact that we do need to remember quite a lot of content as well.
Brilliant. Thank you, Brenda. That's all the open questions we have, although I feel that we might have one appearing in the chat in a few moments. So I'm just going to give our attendee a chance to pop that in. Please, if you're sharp, we've got one coming through in the Q&A now. So this is about students. Should they have any prior knowledge for the syllabus change?
No, I don't think so. Very few students know what's in the course before the details of what's in the course before they undertake it. So no, I don't think there's any need for that. I mean, if they're borrowing books from a previous year, they need to realise that a lot of the topics won't be included anymore. But prior knowledge of the new course itself, no, I don't think there's any need to tell them that, no.
Brilliant, thank you. I have just seen that one more question has come in. Do you think that's a good idea to use questions from A-level biology to facilitate students' understanding?
Yes, I do. I think A-level questions, I think the concurrence between A-level and IB is pretty strong, and I think any sort of question is relevant and gets students thinking and writing the correct amount. One thing I would add, at present, if you use the IB questions which are online, these have not yet been updated to reflect the new syllabus. So you will have to use some of the old questions. I believe it's going to be done before the autumn, but I'm not certain. But any questions that you can use. Another good source is the American examination system too. They also have many useful questions.
Brilliant. Thank you very much. And thank you for all your kind comments that are coming in for Brenda. That's fantastic. So thank you for attending. I'll just reiterate what I said before. Remember that 7 days after the webinar, we'll send you an email thanking you for your attendance with a link to the recording of the webinar and the webinar slides. You can use this email as proof of your attendance. To find out more about the resources discussed today, please speak to your sales representative or visit the links that my colleagues have just been putting in the chat. When the webinar ends, a short survey to complete will open on your screens. Thank you for any answers you provide. Good luck with your teaching and goodbye.
Good luck and goodbye.